Good afternoon, everybody. By just looking at this picture, what you can see that there are two different mice. One is made of paper, and the other one is it's alive. But as a developmental biology, what I can see is this. So one is very easily made. You only have to follow instructions. But the other one is amazing because it's formed by a bunch of undifferentiated cells, and we don't know how to reproduce that. So, as a developmental biologist in my lab, what we try to answer is how, from a bunch of undifferentiated cells, a whole organ arises and it's functional. So, to answer that question, we use Drosophila melanogaster as a model because it has a very short life cycle. We call it takes 10 days. And the juvenile form is it's very different from the adult form. So this is possible because there is a process called metamorphosis happening at this stage, at the pupa stage. So here we have the cuticular structures of an adult wing, an adult fly, sorry. But in order to understand how are these structures formed, we have to recapitulate development till the larval stages, where we find the imaginal discs that are groups of undifferentiated cells that will give rise to adult structures. They don't differentiate during larval stages. They only grow and proliferate, and then by metamorphosis, they start differentiating and they will form the tissue, the adult tissue. In the lab, we are mainly focused in wing imaginal disc. It's this one here that will give rise. The pink part will give rise to the notum, and the blue part will give rise to the proper wing. So, in order to understand how that happens, we have to understand the main characteristics of the wing. As I like to say, the wing is a 3D structure because it's composed of two layers of cells. So, it has three main axes. One is the anterior-posterior axis. The other one is the dorsoventral axis, and because it is a limb, it has the third axis, that is the proximo-distal axis. So, this is proximal, and this is distal, and it only relates of how far from the body trunk the whole structure is. So, these three main axes are set in the wing imaginal disc, and it's a genetic regulation. The anterior-posterior axis is set by the expression of the engril gene in the posterior. Then apterus, the apterus gene is expressed only dorsally. So at the end of this phase, what we have, it's a wing disc that is able to differentiate between anterior, posterior, and dorsal and ventral. And I don't know if you are able all to see me, but in order to get the third dimension, what it has to happen is, if my right hand is does everybody see me? Okay. If my right hand is dorsal and my left hand is ventral, and this is the DB boundary, the tissue starts folding and it everts, it goes out of the plane. So this sheet of cells and this sheet of cells come together. So this is why, you have a 3D structure from a 2D structure. So this is very interesting, and as you know, it's done by genes. So the main pathway we study in the lab is the wingless pathway. It has this pattern of expression here. This is the DB boundary. I don't know if you can see it in red. And then there is a ring surrounding the wing pouch. So it encloses the part that will form the proper wing in the adult. And okay, the pattern is nice, but the phenotype is nicer. Because when you don't have wingless, when a fly doesn't have wingless, it doesn't have wings. So it's very nice. But this process is not so straightforward because wingless effects depend on its level depending. So with no wingless, the fly has no wings, but at low levels of wingless, we have smaller wings without margin. And when you overexpress wingless at mild levels in all the wing pouch, what you get is high proliferative cells that will form a very big wing. But if you put more dose of wingless, what you get is a smaller wing, very differentiated. So mild levels of wingless cause proliferation but stop differentiation, and high levels of wingless stops proliferation and start differentiation. So this is a striking thing, and we know that wingless is doing this, activating different targets that ultimately will control wing development. But these targets are unknown, so we are trying to know which targets are causing these ultimate effects. So, in order to do that, we did microarrays, and after struggling with the eternal list of the bioinformatics gave us, we end up with a lot of genes and 5 main different phenotypes. We got a smaller wings, wings with vein defects, wings that are not able to unfold properly, so they keep folded, wings that lacks the margin, and it also has notches, and other phenotypes like this one that I like it the most because we call it Mickey Mouse because of the shape. This is, for me, it's amazing because the anterior part of the wing is completely messed up. There is no differentiation and there is a lot of proliferation, but the posterior part it's normal, so it's a wild type. So this, for me, it's amazing, yeah. So we are not that naive, and we know that wingless pathway is very interesting to us, but it's only one tiny bit part of the whole gene regulatory network. So we all have to we have to know all this in order to understand how this is formed. And going back to the first example that I like it, only to say that in our way to create our own instruction manual of how development works. I know that this is my thesis and it's very important to me, but in the real world, it's only like I have explained this little fold here. But I hope that it's a very important fold. So only to finish, I want to thank you all the people that are always by my side. Thank you very much.